I hope you can see them. So thank you so much for inviting me to present my work on C. difficile. C. difficile is an obligate anaerobic bacterium that was first identified in the healthy stools of infants. And it was only later associated with a condition called C. difficile infection, or CDI. These symptoms include diarrhea and or pseudomembranous colitis. And the diagnosis is performed based on the presence of symptoms as well as apologies. Also, positive laboratory test. In terms of burden, C. difficile infection is responsible for more than 200,000 hospitalizations per year, and these numbers refer to the US alone. So the first question that I had was, what kind of, what does the gut microbiome in CDI patients look like? And of course, I wasn't the first one to ask this question, but previous studies were either focused on 16S, so had a limited taxonomic resolution, or they were severely underpowered. For this reason, I decided to proceed doing a meta-analysis of all the publicly available metagenomic CDI cohorts, which currently are only 10. For a total of 534 metagenomic samples. We had samples coming from subjects that were diagnosed with CDI, so the CDI samples. We had samples coming from subjects that were diagnosed with other diseases, what we now call here disease controls. And we also have samples coming from healthy subjects, which here we define as healthy controls. So pretty much as we were expecting, CDI samples were characterized by a significantly reduced microbial richness compared to the other sample groups. But most importantly, one thing that I was really interested in was if we could find a CDI-specific microbial signature. And this analysis was performed in collaboration with Jakob Wirbel, also here at EMBL. We trained a lasso model with repeated cross-validation. In this heat map, every row is a bacterial species and every column is a sample. And every, let's say, every color here is assigned based on Z-scores, which are based on the relative abundance of each bacterial species in each sample. And as you can see here, we did indeed find a CDI. We did indeed find a CDI-specific microbial signature that was distinguishable from the signature associated with disease controls and healthy controls. And we had an overall AUC of 0.81. But however, like proceeding tracking C. difficile across the sample groups, we could find only sporadically C. difficile in diseased and healthy controls. But the very interesting and surprising part was actually to find C. difficile in only 30% of the CDI samples. The first potential explanation, of course, that we thought of was that we were dealing with a sensitivity issue due to our metagenomic approach. But we performed different analyses that actually confirmed that C. difficile detection in our case was not significantly limited by the sequencing depth. The alternative explanation is an overdiagnosis issue, and actually there is a considerable body of literature that points to the fact that performing an accurate CDI diagnosis is actually surprisingly complicated and surprisingly challenging, because the CDI diagnosis accuracy really depends on what kind of diagnostic tests are used. And just to give a quick example, the enzymatic immunoassay, which is, let's say, in this specific case, the toxin enzyme immunoassay that looks for the presence of C. difficile toxins, which is one of the most commonly used tests in the clinical context to diagnose CDI, can have a sensitivity that varies from 51 to 94%. Which of course depends on different manufacturers. So there is a huge variability in both sensitivity and specificity. But also very important is to remember that CDI symptomatology is not specific of C. difficile, but there are actually a range of different bacterial species that can cause identical symptomatology, meaning diarrhea and also pseudomembranous colitis. So the next step was to look if we could find any of these species in our CDI samples. So in this linear mixed effect model, you see in blue highlighted the species that are depleted in CDI, and in yellow, the species that are enriched in CDI. And in particular, sorry, in yellow, yes. In red, we highlighted the species that are able to cause a CDI-like symptomatology. And as you can see here, we have Escherichia coli, we have Klebsiella oxytoca, and a bunch of other Clostridia. So in more than 90% of the CDI samples that did not have C. difficile, we found at least one of these species that is able to cause a CDI-like symptomatology. And most importantly, and also very worrying, is that 2/3 of the studies that we included in our meta-analysis did not comply with the recommended guidelines for CDI diagnosis, really stressing the fact that we could be dealing with severe overdiagnosis. But so far, I mentioned, like I talked about C. difficile in the context of C. difficile infection, which is a very specific and limited context because usually it refers to adults and elderly that are diseased, and in particular to their gut microbiome. But I wanted to have a more broader approach, and I wanted to look at C. difficile in other age groups, in other body sites, in other hosts, as well as in other environments. So for this reason, I started building this global survey for C. difficile. I put together around 253 publicly available data sets for a total of 42,814 metagenomic samples coming from 84 different countries. We cover here like several different environments, different body sites besides the human gut, and also a lot of samples from the animal gut. But as you can see, really the majority of the samples are coming from the human gut. And here really we could cover the whole human lifespan from 0 to 107 years of age. In this category, we also included 447 novel gut metagenomes from a citizen science project that was carried on by the Bork Lab. So the first thing I wanted to look at was the C. difficile prevalence over lifetime. And here you can see that it's very clear that in both healthy and diseased subjects, C. difficile is a very common bacterium within the first 4 years of life. And then it becomes a much rarer taxon with average prevalence less than 1% in later stages of life. If we look instead at the microbial diversity that is associated with C. difficile, for example, here, if you focus on healthy adults, you see that healthy adults that have C. difficile have a significantly lower microbial diversity than the age-matching counterparts that did not have C. difficile. And this is valid for both healthy and diseased adults. And actually, it's valid for all the other age groups, with a significant exception of infants. Infants that had C. difficile instead had the opposite trend, so had a significantly richer microbial community in their guts than the age-matched counterparts that didn't have C. difficile. And this was valid, this was true independently of the health status, the geographic location, as well as the gestational age. So it was a very strong signal. But so far, I described the microbial community that is associated with C. difficile in very broad terms. The next step was to actually look at which bacterial species were co-occurring with C. difficile in which stage of life. So in this heat map, every column is a bacterial species, and every row is an age group in both healthy and diseased subjects. And the association of each species with C. difficile is highlighted in orange, in shades of orange if it's a positive association, meaning co-occurrence, or in shades of blue if it's a negative association, meaning co-exclusion. And as you can see, we have two very different groups. We have group one, which is a group that includes species that on average are co-occurring with C. difficile across all different age groups in both healthy and diseased subjects, while group number two is instead composed of species that are co-occurring with C. difficile almost exclusively in healthy infants. Interestingly, in this group here, we have several beneficial species, for example, species that are known butyrate producers. All of these species in group 2 are also obligate anaerobes, and are species that are typically found in the gut of healthy adults. And also, in addition to this, I think very important, but I will not show here the results for time reason, healthy infants that had C. difficile in their guts had a gut microbiome that was much more similar in terms of composition to the gut microbiome of their mothers. Not just any adult, not just any mother, but their mother specifically. And with this, I would like to wrap up saying that C. difficile so far has been studied with a disease-centered perspective in mind, meaning studying C. difficile for its potential to cause C. difficile infection, which means that we are focusing on adults and elderly, which is a very specific age category in which C. difficile is rarely found and is associated with multiple deleterious species that are capable of causing CDI-like symptomatology. And in also this age category, there is a significant concern for overdiagnosis. While on the other hand, I try to advocate for more like a species-centered perspective, where we study C. difficile also in other age groups that so far has been severely understudied, such as healthy infants. And let's not forget, healthy infants, it's exactly the first place where C. difficile was identified in. And in healthy infants, C. difficile is not highly prevalent, is also highly abundant. It is associated with a series of beneficial species, and also with multiple indicators of a healthy gut microbiome development. So overall, in this context, C. difficile doesn't look like a pathogen, but it looks like much more like a commensal. And with this, I also like to highlight that if you're interested in these results, you can find all of these plots as well as many others in this preprint that has been recently posted and is available online. And I would like to thank all the members of the board group, in particular Sebastian Schmidt and also Jakob Wirbel from the Zeller team. And with this, I also would like to thank you for your attention.